You know, DNA and RNA are both made up of the basic component which is nucleotides. However, RNA is single-stranded, while DNA is double-stranded, also known as double helix. Well, and is made of three basic components, which is a phosphate group, a pentasugar, and a nitrogen base, which includes thymine, adenine, cytosine, guanine, and uracil form of RNA. So DNA replication started when an enzyme called DNA helicase goes through and breaks down the nitrogen bond.
A joke that may suit this situation could be, I wish that I could be RNA helicase so that I can insert those genes. Nitrogen base that get bonds is also known as the two guys who are being best friends. The girl which both guys fall in love with would be called a DNA helicase. She would break the relationship between the two best friends since she don't like neither of them.
The point where it starts is called the replication point. The two strands that are splitted are called the leading strand and the lagging strand. The leading strand is started with the 5' to the 3', while the lagging strand is started from 3' to the 5', which is also known as anti-parallel strand. Before leaving the nucleus, cap is added to the 5' end, which is called the 5' cap, and a tail is added to the 3' end, which is called the poly A tail. And this gives the mRNA protection, ease, and it makes it easier to leave the nucleus. This addition is also making it easier to connect with organelles later on. Now let's move on to the RNA replication. So there are two steps in the RNA replication, which are translation and transcription. The first process is called transcription. The enzymes that start this process is called RNA polymerase, and its copy DNA sequences down 3. And its copy is into a similar language type, which is called the messenger RNA, sourced as the mRNA. RNA polymerase stops when it reaches the termination stage.
To explain what is anticodons and codons. Codons, in precisely, the definition of codons is the triplets of nucleotides. It's basically 3 nucleotides grouped together to form a group of 3 nucleotides. Precisely, anticodon is a triplet of nucleotides in tRNA that is corresponding to a complementary codons in mRNA. Let's start with a basic example by matching codons and anticodons together. As you may know, certain codons only matches with certain type of anticodons to make it efficiently working. There are 4 types of codons and anticodons in general: adenine, guanine, uracil, cytosine, and in DNA it's thymine instead of uracil. They are also called the phosphate group. First, to start with AUG, we'll find a triplet anticodons that matches with the following codons. A, which stands for adenine, would matches with U, uracil. So we have A and U. Continue on, we have U, uracil, which alternatively match with A, adenine. Next we have G, which is guanine, it matches with cytosines, which is C. For the rest of the example, which is UCC, AGC, CCU, you will continually do it based on the templates that I listed before of the phosphate group. Let's do a second example. UCC will matches with AGG. Uracil tyrosine, which matches with adenine, planin. The following two, it's just an exercise that you guys can work on. This is an amazing example of what you guys should have do in this codons and anticodons corresponding test or quiz, whatever you may call it.
So after the DNA has been splitted, RNA primers goes down and starts the trail for a few nucleotides. Then the DNA primers take it from there and go through an added matching nucleotide onto the main stem all the way. Now that's only for the leading strand. So for the lagging strand, DNA polymerase can only add nucleotide at its 3' end, while there must be the RNA primers to go in, laying down short RNA primer so the DNA polymerase can have a starting point. This fragment is called the RNA fragment is called the Okazaki fragment. And then after all that, another DNA polymerase has to go through and replace all the RNA primer. Finally, the fragment gets drawn up by an enzyme called DNA ligase.
The leading strand is a parent strand of DNA which runs in the 3 to the 5 direction toward the fork, and it's able to be replicated continuously to the DNA polymerase. While the other strand is called the lagging strand, we can only record it from the 5 to the 3 directions, which made it to record it in a small fragments called the Okazaki fragments. By that, it will be linked together later on by a glue kind of glue linked together called the DNA polymerase. This will result in the two identical strands that look coordinately the same.
The process is called translation. Translation is when an mRNA is attached to the ribosome, and in the ribosome is divided in 3 sites: the P site, the A site, and the E site. The P site is where the mRNA interacts with another RNA called the transfer RNA, shortened as tRNA. The tRNA translates the language of the nucleotide to the language of an amino acid. On one end of the tRNA is the amino acid, and on the other end is a specific sequence of the 3 nitrogenous base.
Each codon along the mRNA strand is read by the ribosome and match up with a corresponding tRNA molecules. tRNA is a long chain of RNA with 3 nucleotides on the bottom, which is known as the anticodons, and the amino acids on the top. Each tRNA has amino acid attached on the top, which will release ribosome and the anticodon match up to a codon. The amino acids that binds into the ribosomes will create a chain of process. tRNA is a dictionary because it translates the information into another language.
But before leaving, the mRNA will go through a process called RNA splicing. The stuff that gets spliced together is called the exon. The thing that gets out is called the intron, which will stay in the nucleus and got recycled for new material. And the mRNA can move out of the nucleus from then. During the translation, there's a step called agglutination. This is when the growing polypeptide on the P site is moved to the A site. When the stop codon encountered in the A site, a release factor enters, translation is terminated. This process is called termination, which is when ribosome is associated and protein is released.
As elongation continues on, new tRNA entering the A sites, and the ribosome that moves along the mRNA, a chain of protein is now flourishing. When the stop codon encounters the A site, the release factor enters the A site. Translation is now terminated. This step is called termination. The ribosome is dissociated. A newly formed protein is now released. See you in the next episode of AS Student News.
Bye.